In glycolysis, ATP molecules are produced from a molecule of glucose, a 6-carbon molecule. Glycolysis takes place outside the mitochondria. To begin the process, 2 ATPs are used to oxidize glucose. Next, the molecule is split in half into 2 3-carbon molecules. Now, as chemical bonds are broken, the molecule NAD+ acts as the electron carrier by picking up electrons and hydrogen ions from the 3-carbon molecules, becoming reduced to NADH. Phosphate groups from the 3-carbon molecules are attached to ADP. Which forms ATP as energy is released from creating NADH. This formation of ATP using phosphate groups that came from other carbon chain molecules is known as substrate-level phosphorylation. These atoms are then rearranged into 3-carbon molecules, which releases water under aerobic conditions. This final reaction is where ADP forms into ATP. From these phosphate groups, the leftover carbon molecules are pyruvate. For every glucose molecule, only 2 net ATPs are produced, while 4 total ATPs are produced in the entire process, along with 2 NADH molecules.
After cytoplasmic glycolysis has converted the 6-carbon glucose into 3-carbon pyruvates, they diffuse into the mitochondria to where they flow to the pyruvate dehydrogenase complex embedded within the mitochondrial membrane. There, the 3-carbon pyruvate becomes a 2-carbon molecule of acetyl and a molecule of CO2 is released. Acetyl is a sugar that will continue cellular respiration and will attach to a protein known as coenzyme A, also known as CoA, by removing electrons, aka oxidizing the acetyl. Acetyl-CoA is then transported to the mitochondrial matrix. High-energy electrons are released and carrier molecules known as NAD are assembled in order to transport these electrons. When the electron binds to the NAD, a molecule of NADH is formed and ATP is released.
Hello, I am doing the Krebs cycle. So the Krebs cycle is the powerhouse of cellular respiration. It takes acetyl coenzyme A, which is produced in the oxidation of pyruvate and glucose. NADH, FADH2, and ATP molecules are transported, produced, and broken down for energy to power cellular respiration. This can take place in the matrix of the mitochondria in eukaryotic cells, while it takes place in the cytoplasm of prokaryotes, sorry. Okay, so our step 1, starting with acetyl coenzyme A. In the first step, it joins with a 4-carbon molecule, oxaloacetate, which we will come back to later, and it releases the coenzyme A group and forms a 6-carbon molecule here. In the next step, citrate is converted into its isomer, isocitrate. This is actually a 2-step process, but I'll get into that. In this step, isocitrate is oxidized and releases a molecule of carbon dioxide, leaving behind a 5-carbon molecule. And yeah, that's during this step. And during this step as well, NAD+ is reduced to form NADH and another carbon dioxide. The 4th step, again, very similar to what I just explained. NAD+ is broken down into an NADH molecule and a byproducts, carbon dioxide and a 4-carbon molecule. Next, we have in step 5, the coenzyme A of succinyl coenzyme A is replaced by a phosphate group. Usually when it transferred ADP to ATP. Now in this sense, sometimes ADP is turned into GDP, and when these two, both the phosphate and the GDP combine, they combine into this GTP molecule. In this next step, succinate is oxidized, forming another 4-carbon molecule called fumarate. In this reaction, 2 hydrogen atoms with their electrons are transferred to FAD, which produces, again, FADH2. This step is very important because it transfers electrons directly into the electron transport chain. Step 7, water is added to this 4-carbon molecule that has been taken into this entire part of the cycle and is made into NAD+. And in this last step, oxaloacetate is regenerated by oxidation of malate, which is another product of NAD+, and the reduction into NADH. And then the cycle just starts back over again. So thank you.
Oxidative phosphorylation is the process in which cells use enzymes to oxidize NADH and FADH2 to create energy. This process includes electron transport and chemiosmosis. Electrons are delivered by NADH or FADH2 to an electron transport chain where they are oxidized, as shown here by this light yellow rectangle. The electrons are then passed between proteins by electron carriers. Electrons are represented by the yellow circles and the proteins are shown in brown. Using the energy that is released from the transfer of electrons, hydrogen ions, or protons, are transferred across the mitochondria's inner membrane, as shown here by the blue circles. This process where protons are transferred across the membrane to create an electrochemical gradient is called chemiosmosis. These ions transfer back across the membrane through a turbine, ATP synthase, to create a lot of ATP, as shown here. This process produces most of the energy in cellular respiration. Then, oxygen takes electrons from the electron transport chain, as well as 2 hydrogen ions, creating water molecules. The orange circles here are oxygen, and it's showing the picking up the electrons, the yellow circles, and the hydrogen ions, the blue circles.
Fermentation occurs during anaerobic respiration when oxygen isn't present to react with glucose molecules, as opposed to aerobic respiration, which requires oxygen. During lactic acid fermentation, a lack of oxygen during the breakdown of glucose results in a lack of transfer of the electrons, therefore reducing the pyruvate instead of oxidizing it. NAD+ keeps cyclically pairing with high-energy electrons from the pyruvate. The lack of oxidation forces a repetition of the glycolysis process, resulting in an accumulation of NADH. A compound known as lactate is formed. In bacteria, instead of producing lactate, the primary product of fermentation is ethyl alcohol. Just like in lactic acid fermentation, alcoholic fermentation involves pyruvate being reduced, resulting in an accumulation of NADH, creating ethyl alcohol. A byproduct of this reaction is CO2.
The alternate scenario I will be discussing is atherosclerosis. As a human ages, the arterial tissues become less flexible, leading to the straining of cardiac muscles. A lack of exercise and a bad diet can lead to the quickening of this process. Fat and cholesterol will build up on the insides of arteries, causing an extremely strained arterial blood flow. Since oxygen cannot get to the mitochondria of cells, oxidative phosphorylation stops. This is because there aren't any oxygens to act as the final electron acceptors in the electron transport chain. This means they also cannot pick up any protons coming from ATP synthase. This also will decrease the amount of energy being produced, leading to a large energy deficit, so now the cells can't function. Since the bloodstream carries oxygen to all cells and oxygen is a reactant of aerobic cellular respiration, a severe case of atherosclerosis will prevent cells from carrying out the process of aerobic cellular respiration. Cells that would experience the immediate effects of the lack of aerobic respiration would be cardiac cells, due to the heart not receiving enough oxygen via the bloodstream. The same effect would repeat for the rest of the cells not receiving oxygen.